Associate Dean of Graduate Studies in the College of Arts and Sciences at the Florida International University. She's also a research associate at the American Museum of Natural History in New York City. Her laboratory has supported several graduate students conducting research in amphibians and reptiles in southern Florida, as well as a variety of sites in tropical areas. To date, she has mentored 7 PhD students and 16 master's students, and has published over 100 papers and contributed to 2 books, including a guide to the Satellite Biological Research Station on amphibians and reptiles. And her research has focused on the ecology and evolution of the strawberry poison dart frog and amphibian reptiles in the Neotropics. And she has also focused on the ecology and defense of poison dart frogs, as well as a variety of studies on the conservation of amphibians. And would you please help me welcome Maureen Donnelly.
Thank you, everybody. It's an honor and a privilege to have been invited by everybody, and I thank the organizers, and I hope I don't leave you disappointed. I want to shift our focus a little bit, and I want to look at One Health through the lens of amphibians. Amphibians are the first and successful group to transition from the aquatic environment into the terrestrial environment. It's an old lineage that has survived countless bouts of the forbidden word, climatic change, over the course of Earth history. The things that wiped out the dinosaurs didn't hurt the amphibians, but now this group is threatened, and 40% of the extant species are under threat of extinction. When you think about that, 7,000 species of amphibians blinking out, it's a little troubling and disconcerting. And I was asked a couple of years ago to present a talk on conservation of amphibians in the New World Tropics, and I agreed to it before I had a talk. And then I had put a talk together, and as I was designing the title, A Tragedy in 3 Acts, good alliteration, it sounded good. But then I thought more deeply about it and thought, what does Shakespeare mean by a tragedy? And a tragedy takes place when the focal hero of the story suffers more dramatically than he or she should by the conditions. And I think by the end of this, I hope to convince you that amphibians are in fact experiencing a tragedy, but I think that through continued research, we can try and stem the tide against this loss, and I want to take us on that journey today. Conservation is very much a collaborative effort. Nobody goes out and does conservation alone. And all of the folks listed above are students or collaborators who have helped me shape my thinking about what conservation is and what it means. And then the agencies, oops, wait, back up. Gotta thank the money. The agencies on the bottom half of the slide are those that contributed the green energy that keeps the research moving. It's hard to do this, and it's hard sometimes to engage, but we keep after it, and we're grateful to everybody who's helped us pursue some of these questions. So the 3 acts of the tragedy are set up. I want to first, for those of you who don't know about the problem, I want to describe the declines of tropical amphibians. I want to talk about research cycles that were initiated in Costa Rica in response to the crisis. And then talk about how we move forward. Because hanging our heads and just saying it's doomed isn't moving along. So we have to find a way to engage and try and solve some of these issues that affect probably not only amphibians, but everybody in the One Health notion. And are they canaries in the coal mine? Probably. So in Act 1 of the story, I'm going to go through 3 scenes: why the tropics, why amphibian declines are part of the biodiversity crisis that's been in our lexicon now for over 30 years. And then I want to talk about some of the applied research that grew out of recognizing we had a problem. So why the tropics? Well, there's lots of reasons for the tropics. 1/3 of the Earth's terrestrial surface is located in the belt that girds our planet. And although we tend to think of the tropics as this sort of luxuriant green hell, if you will, in fact, we know that in the tropics, there's a variety of habitat types, from deserts to treeless paramo to this luxuriant sort of lowland forest that I've loved my entire career. Half of the world's living species live in that tropical belt, which makes it attractive if you're interested in biodiversity. And finally, because so many taxa increase in their diversity as they move towards the tropics, and we've understood this for a long time, there's a rich base of theory around which we can couch our conservation and ecological questions. 50 million square kilometers is what we're talking about inside this tropical belt, and half of that is in Africa. Yesterday we heard great talks from some of the students who have worked there. If any of the youngsters in the room are looking for a place to go do research, Africa is calling out. Basic biology of most species there is unknown, so if you're looking to go make an important contribution, Africa is where you can do it. We can draw these lines of latitudes effectively on the globe, but in fact, organisms follow lines of latitude about as well as they follow geopolitical boundaries. They don't. And for example, tropical forests, mangroves, coral reefs, these are tropical ecosystems. We know they occur in the tropics. Well, they also occur here in southern Florida, which sits outside of that tropical band. How does that happen? Well, warm water from the tropics pulls up along the eastern coast of North America, and so in southern Florida we're graced by these tropical ecosystems that otherwise we'd have to get in the plane and travel south to experience. Almost 60% of lowland forest in the world is located in the New World, tropics. And 30% of that forest, of the world's forest, lives in a single country, in Brazil. So when things start to get complicated in Brazil, as a rule, tropical biologists start to get a little nervous because they hold so much of that forest, not just in the Amazon basin, but north of it as well in the Guianan Shield. If we don't stop what we're doing, we'll lose lowland tropical forest in under 200 years. And the threats are scary because only 5% of tropical forests around the world is protected. And protection, is that good for us or not? I will show you examples later where it's a little bit more frightening because protecting isn't often enough. You can draw lines around a park, but you can't stop the flow of disease organisms. And we'll talk about that as the talk moves along. Paul showed a version of this slide, and I'm going to show it again because the real driver in everything, and the missing element of discussion at the conservation table is this increase in human population growth. And it is still going up. While it may stop in 2050, there's no sign that that's going on yet. And most of this growth is taking place in developing countries that are largely located around that tropical belt. So as these nations develop, as their populations grow, and here's a shot taken just from India because it was the most dense picture I could find, but all of this human growth requires land for growing crops, for growing livestock, and for living. And of course, everybody in the developing world wants to be developed. So they want to have all of the things that they see in the developed world. And as this goes on, as human population growth has gone on, so has the accumulation of greenhouse gases. CO2 is the best studied. It's the most common gas we speak about when we talk about greenhouse gases. It's probably not the most severe of them. My husband is an ecosystem ecologist, and he says methane is where you really need to be looking. But we know that CO2 has increased through time as the human population has gone up. And as CO2 is increased in the atmosphere, the temperature anomaly is also going up. And Paul indicated that by 2050, we're anticipated to see a 2 degrees C increase in basal temperatures. And what this means, of course, is that we have one planet, one One health heating up. And what does that mean for organisms like amphibians and reptiles and fishes and insects and plants that don't have the capacity to regulate body temperature? We don't know what this global change will do to these organisms that regulate body temperature behaviorally through their interactions with their microhabitats. When I was a graduate student back in the Pleistocene, no, it wasn't that long ago, but E.O. Wilson, Norman Myers, Peter Raven started talking about deforestation rates in the tropics and that we were losing species faster than we can name them. And it was coined the biodiversity crisis, and we started to talk about it with some conviction in the late 70s and early 1980s. And I want to talk about the amphibian declines as part of that crisis. Again, where this whole class of vertebrates is affected. And in Costa Rica, the Monteverde golden toad, now known as Ancilius peregrinis, is the poster child for amphibian decline. My colleague and friend Martha Crump received funding from the National Geographic Society to study the interesting reproductive biology of the golden toad. These frogs live in one location, up in the Tilarán Range in Costa Rica in the Brillante Gap. They are underground dwellers that would emerge at the first rainy season rains and have these crazy toad orgies up in the Brillante Gap. Males and females are sexually dimorphic in color. Males are this bright orange color. The females are black with some attractive spots. But Marty received funding at the encouragement of the local community, this is their iconic animal, to study their reproductive biology. Marty got the money and she put a team in the field, and there were 4 toads in year 1. And there were 2 toads in year 2. And in 1989, when she went to the first World Congress of Herpetology, instead of describing reproductive biology of the golden toad, she described the crash of this toad. And at this first World Congress of Herpetology in England, everybody started to talk after Marty's presentation. You know, my frogs, I don't see as many in the Sierra Nevada of California, or at my favorite study site in Australia. And as people began to talk at that Congress, we understood that this was a problem that was global, and a problem that affected the 3 living orders of amphibians, the caecilians, the salamanders, and the frogs and toads. David Wake was instrumental in organizing the Irvine Conference that followed on the heels of Marty's report, and we started to really dig around to see if, in fact, there were true declines that we could differentiate from natural variation in population size. And amphibians are classically known for having variation in their numbers at a breeding pond in a given year. And many people initially suggested that we weren't seeing declines at all, we were just seeing some fluctuation, and everybody should relax. Well, not everybody relaxed, fortunately. Joe Mendelson, who is now at Zoo Atlanta, heading up their conservation program, has likened the extinction of amphibians to the extinction of the dinosaurs. And David Wake and his former student Vance Brindenberg in 2008 in a PNAS paper described this current extinction event as our 6th mass extinction event for the planet. And it's not just frogs, right? It's songbirds, it's corals, it's a lot of things that are sort of blinking out in the Anthropocene, if you will. The conservation threats are the conservation threats that are true to everybody. Habitat modification and fragmentation are the primary drivers, and I'll show a graph that summarizes those data in a minute. All of these things were implicated, and of course everybody was searching for the single silver bullet, right? There is a cause, one driver is affecting everybody, and the hunt was on. And we've searched for all of these at various points in time. I remember when UVB was big. But in reality, all of these factors are interacting with one another in a synergistic fashion. And this is really each of these individual drivers acts before we can design appropriate experiments to move forward. And this is sort of a diagram to show what kinds of multiple, it just takes that previous slide and puts it together. But if we were really honest, we would show lots of lines connecting each one of these drivers because they don't exist independently of one another. This is the slide that was put together by the IUCN and with Stewart, the Stewart et al. Group, where they looked at the number of species being threatened by these drivers. And you can see that all habitat loss is a serious consideration for most of the amphibians that are threatened and non-threatened. So this is the big driver. And again, I would argue that's driven by human population growth. What the Stewart et al. Group did in 2004 was look at all of the data to see how uniform the drivers were. And I will tell you that they're not uniform across the globe in terms of pressures, nor are they uniform across amphibian diversities. So what the group did was identify overexploitation or consumption. Those are big drivers in Asia. The developed world, of course, is losing habitat by development. And then the scary part here are the red ones, the red patterns, because these are what we call enigmatic declines, meaning they're going out, but we don't know why. And those are particularly scary when they occur in protected zones, right? When you lose things from national parks, it makes you nervous because where is the protection? How are we failing? And this is the same, this is the slide, this is old taxonomy, this is pre the Frost Revolution, but it shows the basic pattern that not all frogs are equally susceptible to drivers of loss. So there's variation in how amphibians respond to these challenges. So once we knew we really had a problem, what everybody did was dedicate themselves to try to build information bases. Species list, what is richness like, where is our diversity, what are we facing? And one of the things we did was look at the number of threatened species. And the darker the colors on this map, the higher the number, and you can see it corresponds beautifully with the tropical belt. There's a little exception here in North America, where there's a large richness of salamander species. This will come back a little bit later to the story. But by and large, most of the diversity of amphibians is located in the tropics. And if we look at the mega-diverse countries, they're listed here. China and the US make this list because they're so big, right? It's not that they're particularly diverse, but given their huge size, they get in with this list of tropical countries. And with the exception of Papua New Guinea and Indonesia, all of these are our neighbors to the south. So this is our neotropical diversity, and you can see Brazil is leading, although Colombia, a much smaller country, is right behind it. Ecuador, not too far behind either. And these numbers are probably underestimates because the rate of species description has really gone up lately as taxonomists from around the world are working to name and identify species, again, before we lose them. This is another IUCN map. Again, the darker the color, in this case, the worse it is. And you can see that, oops, wait, that really back up. You can see that there are threatened species all across the globe, particularly in Australia as well. But I want to focus here on the neotropics. I did my work, my dissertation research back when I was just a regular biologist. It was all in this big red dot. Costa Rica, almost all of the amphibians that are threatened. You can see that all along in the Andes, there's a lot of threats throughout the Caribbean, even up into Mexico. And in the news recently, a frog from Ecuador here up in a cloud forest has made the news all over the place, the shape-shifting. Right? This is the shape-shifting Christomantis frog that can change from spiny to rugose as if it were like a slime mold. I graduated, I'm a meat boy, I'm spiny, I'm smooth. But this sort of rapidity of change of dermal architecture is a new thing for a frog living in one of those big red dots. So again, we have so much to learn for these animals and they're under serious threat. So in Costa Rica, I want to focus on what happened in a couple of labs, an outside observer, and then how we took this problem into the lowlands. Because when we all thought it was a problem up in the uplands, we never thought we had a problem in sweaty lowlands. And I want to talk about Monteverde because of the golden toad case. That was sort of ground zero for our understanding of conservation. I want to talk about work done by Karen Lips looking at chytrid, which has been her area of focus, and then I'm going to bring us to the lowlands to talk about what we learned, not looking to be involved in this at all, but we found ourselves rapidly in there. And so Alan Pounds was a student at the University of Florida with Marty Crump. He studied lizard ecology for his PhD. And when Marty left Costa Rica to begin her life as a conservation biologist largely in South America, Alan took over the study of frogs. And the 1982-83 ENSO event in Costa Rica was unprecedented. Nobody had seen drought drivers like that before. And when Alan was trying to explain the loss, not just of the golden toad, but of the harlequin frogs as well, the first paper he published associated drought as the driver of the golden toad crash. So the dry period is what caused the decline. And he later connected with some hydrologists and some climate modelers and changed his opinion slightly. It wasn't that drought was just the driver, but rather it was the shift in how cloud water was deposited on the forest. The shift in hydrology was then the driver for the same decline that once used drought. And then in a very provocative publication that came out in Nature in 2006, Alan suggested that climate change was actually the driver of disease dynamics. And this, of course, was quite alarming to everybody who was reading the article, but that article, in fact, focused on one lineage of one family of amphibians, on the genus Antilochus. So, meanwhile, well, Alan's he's the champion of the climate explanation. Karen Lips was focusing on chytrid because she was in Costa Rica at ground zero, seeing mass die-offs and mortality in the south. Karen described first the decline of this montane herpetofauna where she had done her dissertation work, and then was involved with many, 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 many studies, including the Antilochus paper that Alan Pounds was part of. So two labs had the same data set, but examined that data set a little bit differently. And where Alan described climate as the driver, Karen said disease is the driver. Climate has nothing to do with it at all. And later she contrasted disease spread versus climate change in her wave spreading hypothesis, where she describes the spread of chytrid as a wave moving from points of origin. Now, interestingly, so these guys are battling it out. These labs are beating heads over each other, as labs often do. Meanwhile, a young ecotoxicologist that was hired at the University of South Florida, Jason Rohr, published a very interesting paper in PNAS, where he had no dog in the fight. So he took everybody's data and examined everybody's hypotheses using slightly different statistical techniques. And what he found, this climate-linked epidemic hypothesis, what Jason found was that the same thing everybody finds when you look at correlation. Many things can correlate. It means nothing about causality. Lots of things were correlated with decline, including the rate of beer production in Costa Rica over the interval. So again, caution. If you see a relationship, that's great, but it doesn't necessarily mean one is a driver of the other. So always be careful with that. He further looked at this hypothesis Alan put forward in the Nature paper that there was a thermal optimum hypothesis that Alan put forward, but he only used 3 of the temperatures for BD growth. When Jason incorporated all 7 temperatures at which they looked at BD, he got the opposite result. So be careful if you're going to cherry pick data. Make sure you pick it so that nobody else will find out that you picked it wrong. So try not to be disingenuous whenever you can. And lastly, he examined this spatiotemporal spread hypothesis Karen put forward. He found some support for it, but he was equivocal on the drivers. He didn't exactly buy the argument. So we're still thinking a lot about all of these things as we move forward. Meanwhile, oh, and let me speak now for a minute about chytrid, in case you don't know about this organism. Chytrid is, quote unquote, now an emerging pathogen. I'll show you why that's in quotes in just a moment. It was discovered in a zoo population. The National Zoo in Washington, D.C. had a population of Dendrobates azureus, the big, beautiful blue poison frogs. And they were dying in captivity, and they couldn't figure out why. They got a hold of Joyce Langford, who's a mycologist up in Maine. Joyce identified the chytrid and named it Batrachochytrium dendrobatitis. I will call it BD from now on. I'm not going to stumble across all those words. It's a basal lineage within the radiation of Chytridiomycota. These are fungi that are in streams that are normally breaking down coarse woody debris. So that's their typical role in ecosystems. They identified many unique features of BD. They were able to fulfill Koch's postulate. And if you're not a microbiologist type and don't know what that means, it means you have a healthy animal, you add the infective agent to see if it causes death. And in fact, they did fulfill Koch's postulate with BD. And again, I'll talk a little bit about Jamie's work because she examined how animals die from infection. Again, the organism gets embedded in the skin of amphibians, and it goes through this growth and development phase. Once the cap pops out, the zoospores are released, and it's believed that largely aquatic amphibians are affected because the zoospores are presumed to be swimming around in the water. So animals pick up the infection in the water, again, supposedly. I'll show you in a minute why that might not be so true. The fungus attacks keratin, a protein that's common in the skin, hair, and in tadpoles, it's very common in the mouth parts. So you can see where normally in a healthy tadpole, these structures would be dark black from the keratin, as would these labial tooth rows. The tadpoles are not killed by chytrid, but they're clearly reservoirs, and they can keep the organism in the system because it lives on their bodies. Adults, though, don't do very well at all. This is a mummified frog from Australia. If any of you know what frogs do, this is not what they do. This is not a typical posture you would ever see a frog assuming in any way. And again, Forrest and Roberta were on one of these fronts to actually see the decline take place at their site, and they found dead frogs floating in the environment. And again, this is just not something you typically see. Jamie was very interested in understanding how did this fungus actually work? How did it act to cause death? And she worked in Australia in Ross Alford's lab with the green frog, Latoria ceruleus, seen here. And what she found was that basically plasma electrolyte balance went to hell in a handbasket. Loss of potassium, loss of sodium, and basically frogs were dying from cardiac arrest. They have heart attacks because their electrolyte balance goes to hell. And we think what happens, amphibians have a very highly vascularized patch of ventral skin known as the pelvic patch or the drape patch. It's a vascularized area that amphibians will sit down against a moist substrate or in the water and actually pull water up into the skin through that patch. And we think that once an animal is affected by chytrid, there's a disruption of that pelvic patch and an inability to stay in a proper water electrolyte balance. And this nice little life cycle shows how the business works. The zoospores are released, they move around in the water, they get into the skin, they grow up. From the time of infection to the time of death is about 2 weeks. And although the critical temperature when this figure was made was 23 degrees, we know there's a range. It's about 17 to 25 degrees. So people are going, well, wait, maybe this warming world will be okay. Maybe we'll wipe chytrid out. color to be warmer. I mean, we just don't know. It's not a thing we can predict well. What we do know about chytrid is that not all strains of it are equally infective, and not all amphibians are equally susceptible to the disease. Just to show you how rapidly this field changes, a paper was published by an Australian group very recently where they found BD in rainwater. They found the spores in rainwater. This means that if those spores can be picked up and moved by weather, aerial transport of this disease might explain how amphibians that don't use water for reproduction are infected. Because we have terrestrial frogs that we know get chytrid. Additionally, when I say an emerging pathogen, chytrid has been identified from the skin of amphibians housed in collections from 1888. So chytrid has been around for a long time. In the 1940s, there was a huge incident suggesting some event took place, and then certainly now we would argue that a new event has taken place. This slide is also marvelous because it shows the value of natural history collections, and why we keep them, why we have them, and why we marvel at formalin, and how good of a fixative it is, because it lets us go back in time and ask questions historically about the prevalence of disease. And then, again, sort of explaining how some animals are susceptible and others aren't. This paper that again came out 2 days ago, on the 25th of March, by Bruce Waldman and his collaborators, found that susceptibility to the disease is determined in part by MHC genes. And this follows on work that Anna Savage did with the baby Japapiensis, where she found that some MHC complements did better. So if you're trying to manage the disease, you can maybe breed those frogs together to try and reintroduce them to the wild. So the more we know, the better we can be as managers as we try to mitigate the disaster. And the thing that has everybody frightened currently, of course, is this new disease that recently has been identified from Europe, BS, I think it's fitting, is this disease that's been isolated from newts in the Netherlands. It's essentially wiped that group out. Again, it causes serious skin infections. It causes mortality in half the time. And it operates at a cooler temperature regime than does BD. This organism is tied to the pet trade. Salamander pet trade has been linked to BS. Similarly, a lab in Australia has linked the hypervirulent strains of BD seen in other creatures to the pet trade as well. So perhaps there's something about pushing animals in contact, holding them in captive conditions, lets the disease run wild, because people aren't monitoring that, right? They're monitoring the animals that they're going to go out and sell in pet stores. So this is a new emerging disease that has everybody nervous, especially if it gets into the U.S. And the way it could get into the U.S. is through the live pet trade, when Asiatic fire-bellied salamanders are bought by the hobbyists and they get tired of them, throw them outside, and there you go. So once again, we have a capacity to introduce disease, and several biologists are working closely with Fish and Wildlife to try and get them to establish prohibitions against the importation of salamanders into the country to stop the spread. And so meanwhile in the lowlands, this was a mountain problem. Amphibians were dying up in the mountains of Costa Rica in Central America. And we were sitting in the lowlands enjoying the hot, sweaty life, warm temperatures, and there were frogs. We could go find them in the field. We could go find frogs breeding in ponds, and we're sitting there thinking life is good. And then, yes, exactly, somebody mentioned that I've spent a lot of time studying this organism back when I was just a biologist. I was at La Selva trying to understand how territoriality function. We're interested in how these animals acquire their toxicity through diet. So La Selva was a place where I was busy just being a biologist. And the first student in my lab to work on a conservation-related question was Kristen Bell. She was the niece of Ted Stiles, who some of you in the bird world may know from Rutgers. He was a longtime La Selva researcher. And Kristen, as a child, went to La Selva as a kid. with her Uncle Ted. So by the time she went through school and she was a tip student that went to Costa Rica, she spent a semester abroad with OTS, and when she came to my lab, she was very focused on trying to understand how fragmentation of this kind of forest affected amphibians. This is a map. Here's La Selva. It sits at the end of this. If you go up here, this is Volcan Barva. So while this looks like a nice long chunk, it's actually a chunk that's moving up an elevational gradient. So the low part of this is about this much of the preserve. And as you can see, the landscape of Serapequi, now when I first started going there, back in the long ago day, it all looked more or less like this in the zone. And now, of course, it's fragmented quite extensively as human population growth in the zone has grown. We use GIS to examine the variation of fragment size across the zone so we can pick sites for Kristen's study. She ended up selecting 9 fragments in Serapequi. This depended, of course, on landowners giving us access to their patches. We had to negotiate that. We had to negotiate cops shooting at her at the night and all kinds of other things. She persevered anyway. She compared what she found in these fragments using standard techniques to what she found at La Selva, and we got a nice paper wherein we argued that these little tiny patches of forest, at least for amphibians, could be important. 80% of the diversity of La Selva was captured in all 9 of these very small fragments. So the arguing was that these small forest patches can be valuable for amphibians. And Kristen, again, used 2 standard sampling methods. One of them is litter plots. Herpetologists are good at laying a square down on the forest floor. And then you move the leaf litter and horsewoody debris out of the way and capture all the animals that are contained in that square. That gives you a density estimator, which is a good value to go on. And when Kirsten was working up her data, she came to me and said, you know, Mo, maybe there's amphibian decline. I said, well, what do you mean? She said, well, my numbers look low compared to your numbers. And so Kristen was sure we were seeing some amphibian decline going on. And her lab mate, these 2 started at the same time, Stephen Whitfield, said, oh, Kristen, no. We're looking at the naturally occurring variation in population sizes. There's no decline. And so they came to me and said, well, what do you think? I said, well, I think you'll get all the data you can get, and let's try and address this question. So Kristen got a job right away, and she went away to go work for a conservation NGO in New York State, and she handed the files to Stephen Whitfield, who took this project on for his dissertation. He worked up the analysis of the data, and we had 35 years. A 35-year window of data gathered using the exact same method. This is the longest living data set for amphibians in the world, and it's done. Mason Ryan just published a paper that probably has the 2nd longest data set for amphibians in the New World Tropics. We collected, as people do these litter plots, and the leaf litter mat, leaves fall off the trees. The litter mat accumulates during the dry season. Once the rains come, that breaks that mat down. As the breakdown begins, the insects flush like crazy. So the animals that live in this litter mat are feeding, breeding, and living inside that thick mat of litter that covers the forest floor. So as we were trying to sort out the possible drivers of amphibian decline, we could look at the lizards to try and help us understand which hypothesis might explain our data. And then we had 2 data sets. One from patches of old-growth forest and data from abandoned cacao plantations. When Les Holdridge acquired La Selva back in the 1950s, he planted chocolate trees along the alluvial terraces. Once OTS acquired that land in 1963, those cacao plots went abandoned. The trees were standing, but they were no longer being worked actively. They're great places for students to do sampling, and so we continue to gather data from this abandoned agroecosystem over the interval. What we found was frightening. The 17 most common species that you see in the leaf litter over the interval declined. All 12 amphibians, 5 pine reptiles. Everything in old-growth forests declined over the interval. And the decline rate's about 5% a year, which is not nearly as dramatic as a mass die-off. And unless you're sampling regularly, it's hard to detect that change. We were incredibly depressed when we saw these data. When we looked at the cacao data, it got worse. Because 4 species, 1 frog, this beast, Oophagopomelio, my little guy, and 3 species of lizards were static or did slightly better over the same interval. A conundrum presented itself, where no matter what we came up with, right, we had to simultaneously explain decline in one habitat and increase or stasis in an adjacent habitat. This resulted in excessive ethanol consumption by the members of my lab group and tearing out our hair as we tried to sort of face this issue of what does this mean? So what I want to do now is examine each of the explanatory hypotheses for amphibian decline and look to see how our data answers those one way or the other. We concluded in our paper that this natural variation, it could happen, but it's very unlikely that it would happen synchronously for 17 species across the interval. The strong synchronous decline across a variety of taxonomic lineages suggested to us that some external driver is at play. So we ruled out this variation business. We also ruled out fragmentation, because if fragmentation were really a problem for the region, for La Selva, then the cacao plots and the forest plots should have been similarly affected, and we have to throw fragmentation out. Similarly, pesticide drift in the zone, we know that there's intensive agriculture of bananas, Pehibaye, piña, all of these are, pineapple, these are all crops that require intensive inputs of agrochemicals. The drift is from east to west as the trades blow through. It explains the declines we see in frogs, lizards, and in birds, but it can't explain the cacao, right? If things are going to die in the forest, they should be dying in the cacao too. Chytrid, likewise. And what we did to try and get some sense of chytrid, we took some of the animals that we sampled, we had swabs, we sent them off to the diagnostic lab, and for those 24 animals we sent swabs in for, all of them came back negative for chytrid. So we said chytrid isn't present in our sample. And we know that chytrid doesn't, at least we think, today, chytrid doesn't affect the lizard. So we had to toss chytrid as the explanation for our results as well. And what does that leave us with? The forbidden words in my state. And I am a state employee. That means I work for the government. Rick Scott, climate change appeared to describe perhaps what we were seeing. Over the interval of 35 years, La Selva has gotten warmer, as indicated by an increase in the daily minimum temperature. And over the same interval, the number of dry days has decreased, meaning that it's gotten wetter, right? So all other studies that were done looking just at this litter community, we know that the abundance of animals is very positively correlated with the depth of the leaf litter. Deeper the litter, more animals. It's a packing issue, right? We know also that litter decomposition rates are going to be affected by temperature and moisture, and if it's warmer and wetter, the rates of litter decomposition should go up, right? They should be more rapid. And so we think perhaps that in old-growth forest, higher rates of litter decomposition might explain those forest trends. Chocolate trees, if you've never seen them before, they're adorable, but they're nuts. They flush leaves and drop them maybe 3 to 6 times a year. So there's always litter in the cacao groves, always. Even at the height of the wet season, there's a thick amount of litter. And so what we suggested that perhaps litter dynamics might be an explanatory variable for our weird results, with the decline in one habitat and an increase or stasis in the other. And what we concluded from, and we put this forward as a hypothesis to be tested. And we concluded that the lowlands are very much part of the mix in the One Health mode of thinking about things. Just because you're in the lowlands or in the desert or in Galapagos, it doesn't, you're still a part of the N of one issue of one planet. I definitely believe that these gradual declines are harder to detect than a massive crash. It's harder to see it, and it takes more intensive field work to gather the data to demonstrate it. I think this is sort of an understatement. We really don't understand these faunal declines very well at all. And again, the saddest one of all is that habitat protection in and of itself may not be enough to solve the problem. So what do we do besides consume more? Well, what we've done is more research. And I want to encourage all of you sitting out in the world as you think about One Health, you're our future. All the youngsters in the room. You're our future. You're the ones that are going to be the drivers of the solutions to these problems going forward. So I want to talk about some of the continued research on declines in this last section of my talk. This is all work being done by my students just in Central America. And then I'm going to talk about some of the ex situ conservation efforts that have been part of the Amphibian ARC plan. Because, again, if we know chytrids in an environment, can we ethically put animals back into a poisoned world? That's sort of the dilemma we face. So we need to understand husbandry and how we might try and save some of these species that are threatened. So I'm going to talk about Stephen's litter addition removal experiment. He actually tested the hypothesis we put forward in that PNAS paper. I'm going to speak a little bit about what we've learned about disease and pesticides to sort of set up Tyrone just a little bit. I'm going to talk a bit about Justin Nowakowski's fragmentation work, and then I'm going to briefly touch on what my new students are doing in the interest of time, talk about Michelle's work in secondary forests and Luke's testing of release strategies to try and see if we can improve reintroduction. So again, Stephen designed a study. It was a block design. There were 3 blocks in the forest, a litter removal plot where we took leaf litter, leaf by leaf, and added it to the litter addition plot in the block. Those 2 plots were compared to a control. And we followed these plots from the time the experiment was put in place for 16 months. And what we discovered was that not all species responded in the same fashion to the supplementation, but the species that had the most rapid rate of decline over the 35-year window responded favorably to the supplementation of leaf litter. So we argue that leaf litter dynamics are important, and perhaps if it means creating artificial leaves to get them into forests that are losing leaf litter, that's a conservation strategy we might try. What we found, we went back to ask the chytrid question in a more rigorous way. And Stephen led this effort. Again, this is collaboration across a lot of people. But we sampled frogs out the selva for chytrid fungus for a 12-month period. And what we found is that indeed chytrid is present at the selva at low rates, but at the end of the rainy season, which extends about 9 months of the year, during the rainy season when temperatures are lower and there is chytrid. Chytrid is prevalent, and you might envision if you had an abnormal weather year where cool conditions were extended a couple months out, chytrid might become a bigger issue at a lowland site. While we were surveying for chytrid, we also found ranavirus for the first time in Costa Rica, and worse, we found that many individuals were co-infected with not just chytrid, but with chytrid and ranavirus. So these are animals that are challenged, and how these challenged animals respond to climate change or respond to pesticide is a whole other issue that we hope to get to. We also did a study, DDT. We all know it's banned in the developed world. Well, they're selling it. They sell it legally to developing countries all around the world. And you can walk into any ferreteria in Costa Rica and walk up to the shelf of agrochemicals, which is what we did. laid down $200 to buy everything off the shelf, and then we subjected the red-eyed tree frog tadpoles to the recommended application of these pesticides. We read the directions, made the juice, and exposed tadpoles to these things, and found not only was there a huge gap in our knowledge about how pesticides work with tropical species, we found that all of them were horrific for the red-eyed tree frog. Tadpoles had rapid mortality, they did not do well at all, and basically our paper was a call for more research in the area. Immediately, the chemical company people came back to write a letter saying, oh, pay no attention to those people down in Costa Rica. We know everything we need to know about ecotoxicology because we studied the chub. So a fish, these guys actually thought we were going to buy that. A fish species would be enough to understand 7,000 other species that are terrestrial. So we jumped right back in the ring and rode back saying, oh, come on, don't be so nice. So there's lots of work that remains to be done in this arena. Justin Nowakowski, who just recently graduated from my laboratory, took fragmentation studies to a brand new level. He worked with a former student of mine, James Watling, and we conducted a major meta-analysis to understand that you can look at your patch, but the habitat that surrounds your patch matters. In other words, the matrix is important and you need to think about it. Justin actually did experiments to look at resistance, how animals move through different kinds of environments, and looked at the value of remnant trees in pastures. These are student-led projects that Justin organized. And importantly, he used these resistance surfaces, which he obtained experimentally from his field work, to understand connectivity for amphibians. And then I think a really important part of Justin's work is that he brought he used population genetics to try and understand how frog populations might respond to these changes driven by humans in the zone. Most of what we see in the world these days are not primary old growth forest anymore. We're looking at second growth forest, particularly in Costa Rica where government subsidies for cow production fell off. All these pastures have reverted to second growth forests. They dominate tropical landscapes. My colleagues were urging me to work with secondary forest herpetofaunas, and I ran like the dickens to the deanery. But fortunately, Michelle Thompson, one of my students, wanted to work in secondary forest to understand what amphibians and reptiles are doing there. She has 2 chronosequences. All of her study sites were former pastures that are now 0, 5, 10, or 25 years or so older. She has replicate plots in both northeastern Costa Rica, at La Selva, and in the Osa Peninsula to try and understand how amphibians and reptiles are making it in these human-created landscapes. And then I want to finish with Luke Linhoff, who is very much involved in amphibian arc activities. There's an amphibian conservation action plan that was adopted in, I think, 2009. And the amphibian arc is this ex situ group of this amphibian conservation action plan. And I need to wrap up. The amphibian arc involves lots of IUCN groups and the World Associations of Zoos. And they came together in part because of Karen Lips's work in Panama saying, look guys, if all we do is publish papers and we haven't saved a single species, then we're doing the wrong thing. So a group went down to Panama, they went to an intact forest site, they pulled all the animals out because they knew chytrid was coming. And then put them in arcs to try and figure out how to perpetuate them, how to keep them alive until we can learn how to mitigate the disease. And Luke, who came from a zoo environment, is very much interested in experimentally testing release strategies to see how we can help amphibians re-inhabit the world. And so basically, in these ARC facilities, and they exist now all around the developing world, we're basically holding on to animals in the hope that we can right this wrong, at least for this group of organisms. And I hope we do, so that future generations, particularly in the developing world, can appreciate the magic of these remarkable organisms. And with that, I thank you.